Hey everybody, hopefully you had a chance to watch the PowerPoint put together by Mr. Powers on DNA replication. This is here to kind of help you out a little bit. I've always found this animation really helpful from my understanding of DNA replication, and hopefully it'll help you too. You can check out the animation. I put a link to that in Teams as well. The challenge is it's a Flash-based animation, and some browsers aren't supporting that anymore, but I'd like to walk you through it all the same. So here it is. So I'm going to click refresh on this, and what you'll see is this animation starts with a very zoomed in version of DNA, and then it zooms out a little bit. So let's go ahead and watch that process. So here's our DNA zoomed in, and it zooms us out. It starts where you see every single atom, the hydrogens represented little white dots, and everything else is different colors. But here as it zooms us out, we see the basic structure of DNA. Here's the less zoomed out section over here. Again, when I hover over it, I can see all of the different atoms. You can see when I hover over it, the pink ones represent the phosphate bases, and then the yellow or orangish ones represent those deoxyribose sugars. And the bases come across from there as the rungs. You can see them with their individual hydrogen bonds. And so again, you can see, on any of these different sections, they've got the different color codes at the top so you can see which represents which. As we all understand, hopefully at this point, if you have an A on the left side of your DNA, you have a T on the right side, C on the left side, you have a G, and vice versa. So if you have a T on the left, an A, and a G, you get a C. And they're all represented here in this tiny little zoomed-in model. So when we actually go through the process of replication, we have this incredibly long strand of DNA, and this enzyme helicase comes in and unzips or bubbles sections of our DNA. And so you'll see there's the bubbling happening. On the right-hand side of this model, you can actually see it shows you an even more zoomed out section. And it's important to understand this bubbling of DNA for replication doesn't just happen at one spot. It's not like it starts at an end of the ladder. It does it in multiple spots down the DNA strand in order to be more efficient. So it doesn't just bubble at the one end, it bubbles at different spots throughout. So you can see here it represents 3 bubbles of DNA going down. So helicase unzips the DNA, and we have these bubbles, and at this point we can start building in the new strands of DNA. And when we do it, it actually gets started with this thing called RNA primase. It's necessary to have this little RNA strand to get the process going. Eventually that falls off and you'll end up with just DNA, but the RNA primase gets the process going. That's important when we talk about evolution later on, but for now it's just something I'm not expecting you to remember. And then the DNA starts putting itself together. Again, there are a couple of other enzymes involved in this. DNA polymerase also comes in and starts the process. Now what I want you to look at is just this one side of the DNA molecule as we start replication. And you'll notice that the DNA replication works down the right-hand side just fine, where it drops in letter after letter as it pairs up the right pairs. So again, the right side is simple. Your DNA just goes right on down and reads just fine. The problem is DNA can only read from 3 to 5 prime. It can only read and copy in one direction. So you'll notice this part here is getting stitched in 5 to 3. The other side here is 3 to 5, right? So here's our zoomed in model. This side of our DNA is 3 to 5. So it can only stitch going this direction, going from 3 to 5, putting a new 5 to 3 in. The other side, that doesn't work. It can't go straight down the molecule as the bubble works its way down. What it has to do is move backwards in chunks. And so again, if you watch this side, it moves down perfectly clean, goes straight down. The left side here has to form backwards in chunks. So if you watch, again, here we've got our process. There's RNA primase, DNA polymerase. And when I click play again, you'll see this side goes straight down. But this side has to come backwards in chunks. So you'll see the DNA fills in from the bottom to top, again going 3 to 5, going this direction, and then does that over and over again. So here you go. Yep, there it comes in a chunk. And then here comes the next chunk. There it is. And so on and so forth. So since DNA only reads 3 to 5, that back side or that back section has to fill backwards in chunks as the enzyme moves its way on down. What we call these chunks are Wazaki fragments. And they're a little bit different. In the end result, it all looks the same. You get 2 perfectly equal strands of DNA, each with an old and a new strand with it. But on the one side, it fills backwards in chunks, and the other side, it fills in just one direction. So let's watch it one more time. So here we have our zoomed in DNA. Zooming out a little bit, the process starts with helicase forming bubbles down the DNA chain. A little bit of RNA primase gets the process going, followed by some DNA polymerase. And then we see the actual replication, moving 3 to 5 just fine down the one side, the other side filling backwards in chunks with what are called Wysocki fragments. That's DNA replication. You're expected to make a model that looks something like that using some stop motion animation, PowerPoint, or whatever it is you think is best.